In this video we will discuss about lymphoid organs of immune system. Good morning. Welcome to YouTube channel. Bioconcept. Learn with concept. The body organs which are involved in immunity and immune response is called lymphoid organs. These lymphoid organs have some importance in immunity, like this immune cells formation and development. In lymphoid organs, antigens are encountered with immune cells and shows immune response. Immune organs can be classified in three groups, based on function: the primary lymphoid organs, secondary lymphoid organs and tertiary lymphoid organs. In primary lymphoid organs contains types of organs involved in blood and lymphoid cells, formation and maturation. Bone marrow and thymus are primary lymphoid organs. In secondary lymphoid organs, a type of organs in where antigen are encountered with immune cells and immune response occurred. Immune cells can also store in these organs. Spleen, lymph node, mold, gall and ball are the examples of secondary lymphoid organs. Tertiary lymphoid tissue are the infection or where actual infection occurred and immune response initiated. Inflammation in damaged skin. The bone marrow is site of hematopoiesis. In this site blood cells formation occurred. Bone marrow contain hematopoietic stem cell, have properties of self-renewal, and differentiate into all blood types of cells. Most active bone marrows are present in long bones, hip bones, and sternum. This bone marrow also contains some other types of cell in adult stem cell niche. These cells coordinate in hematopoietic stem cell development. These cells are osteoblast, endothelial cells, reticular cells, and sympathetic neurons. Osteoblast are blood-forming cells and regulate hematopoietic stem cells differentiation. Endothelial cells are blood vessel cells, also regulate hematopoietic stem cells differentiation. The reticular site connects blood vessels to bone. Sympathetic neurons regulates the release of blood cells from bone marrow. Bone marrow microenvironment can be divided into two areas: endosteal niche and vascular niche. The endosteal niche is the area which directly surrounding the bone and in contact with bone producing osteoblast. This endosteal niche are occupied by quiescent hematopoietic stem cells in close association with osteoblast. The vascular niche is the area where blood vessels are present in the contact with endothelial cells. These are occupied by hematopoietic stem cells that have been mobilized to leave the endothelial niche to differentiate and circulate. The other primary lymphoid organ is thymus. T cell differentiate in bone marrow and circulate to blood. Then T cell maturation is happen in thymus. Immature T cell is also called thymocyte. Thymus gives specific environment to mature and generate specific T cell receptor on immature T cells. Thymus are reducing its size with age, and adipose tissue and a realer connective tissue takes place of it. In structurally, thymus is bilobe structure, which are located between mediastinum, between sternum and aorta. These two lobes are connected to each other by the connective tissue. Thymus are covered by an outer layer called capsule. This capsule have extensions, which is known as trabeculae. Internally, thymus are divided into two regions. Outer is cortex and inner is medulla. Outer region is also called thymic cortex, and this region contains some types of cells: T cells, scattered dendritic cells, epithelial cells and macrophages. T cells in cortex region are immature and mature both types. The cells have no any receptor on their surface are called double negative T cells, and have both CD4 and CD8 receptors are called double positive cells. Maturation and selection takes place in this area by cortical thymic epithelial cells. In inner region called medulla, the medulla region have single positive T cells, epithelial cells, dendritic cells and macrophages. And here is also cluster made of degenerated flat cells, called thymic corpuscles. Medulla region contains more mature T cells, which have only single type of receptors on their surface are called single positive T cells. Then maturation takes place through medullary thymic epithelial cells. Then this T cells leaves thymus and moves to infection area to encounter with microbes.